Breakthrough: Advances in Production of Retinal Cells for Treating Blindness. I'm Steve Bolero. Welcome to Breakthroughs. Researchers at Karolinska Institute and St. Erik Eye Hospital in Sweden have discovered a way to refine the production of retinal cells from embryonic stem cells for treating blindness in the elderly. Using the CRISPR-Cas9 gene editing, they have also managed to modify the cells so they can hide from the immune system to prevent rejection. The studies are published in the scientific journals Nature Communications and Stem Cell Reports. Age-related macular degeneration of the eye is the most common cause of blindness in the elderly. This loss of vision is caused by the death of the photoreceptors, the rods and cones, resulting from the degeneration and death of the underlying retinal pigment epithelial, RPE cells, which provide the rods and cones vital nourishment. A possible future treatment could be to transplant fresh RPE cells formed from embryonic stem cells. Working with colleagues at St. Erik Eye Hospital, researchers at Karolinska Institute have now found specific markers on the surface of the RPE cells that can be used to isolate and purify these retinal cells. The results are published in Nature Communications. The finding has enabled us to develop a robust protocol that ensures that the differentiation of embryonic stem cells into RPE cells is effective and that there is no contamination of the other cell types, said Principal Investigator Frederick Lehner. We've now begun the production of RPE cells in accordance with our new protocol for the first clinical study, which is planned for the coming years. One obstacle when transplanting tissue generated from stem cells is the risk of rejection, which occurs if transplantation antigens from the donor and the patient tissue differ. Research groups around the world are therefore working on creating what is known as universal cells, which ideally will not trigger an immune response. In a study published in Stem Cell Reports, the same group at Karolinska Institute created embryonic stem cells able to hide from the immune system. Using CRISPR-Cas9 gene editing, they remove certain molecules, HLA class 1 and class 2, which sit on the surface of the stem cells as a means by which the immune system can identify them as endogenous or not. The stem cells lacking these molecules were then differentiated into RPE cells. The researchers have been able to show that the modified RPE cells retain their character, that no harmful mutations appear in the process, and that the cells can avoid the immune system's T cells without activating other immune cells. The rejection response was also significantly less and more delayed than after the transplantation of regular RPE cells, the surface of which still possess HLA molecules. That's amazing. That's amazing. I'll leave a link in the description to the full article. And as usual, please like, share, subscribe, and take care.